Okay, two of your colleagues asked me to go through this and to explain what's going on and try to help understand where these symbols came from. They've gone to a different source. They came up with some problems. They're doing excellent studying. They're finding different examples to try to understand this stuff. So we have these two parents. One is big Y, big R, big R. This has little y, little y, little r, little r. So we have to then assume that they're on different chromosomes. So we have here this parent. Let's say this one's in blue. Big Y, little y, little y, little r, little r. So you have this chromosome. They have this chromosome that has a little y. There's two copies of this chromosome, right? There's two copies. So you can have two possibilities to give it. Now, after meiosis, these would all be in cells, right? So this would be, let me go ahead and create the meiotic, the sperm and the eggs, or in this case, the pollen, then whatever in the case may be for a particular species. But let's make the gametes, right? So this gamete for blue, the gamete would be one possible gamete has a little y in it, right? Right? This other gamete here has a little y. Then this gamete, I'm talking about the parent generation right now. Then this parent here, the little y also has a little r. This also has a little r. All on the chromosome, right? It's all on a chromosome. They're all copies, versions of the gene. Another gamete has another chromosome with little y, little r. And it doesn't matter they're crossed over because they're all little y, little r. And the same thing goes with the other parent that has big Y, big R on one of its chromosomes, right? And it has all of their gametes have big Y, big R. They can all give one big Y, one big R. That's all these cells can give. And the reason it's the only thing they can give is because they're all purebreds at this stage. This first parental generation, this F1 generation, is they're all big Y, big R. They're all one. They can only give one possible gene combination because that's what they have. One big Y, one big R. Right, in each of their gametes. But you see that these gametes go through a cross, okay? And they produce an F1 generation. And this generation is no longer homozygous. These are heterozygotes. Now things get a little more confusing, right? So let's go ahead and delete these and see what's going to happen. Oh, jeez. So now we have two heterozygous parents, a male and a female. And they have these genes. And there could be crossing over, right? We don't know where the crossing over is going to happen. We don't even know if Y and R are on the same chromosome or not. They might be on two different chromosomes. We have no information about that. But we have two parents. One's a male and one's male, and one's a female. And what we do know is that the male has the problem, and both male and female both have this possibility of either giving a big Y or a big R, a little y or a little r. You give any one of those because there's 4 gametes in each of these, right? 4 gametes. And there's 4 possible combinations that can happen because of crossing over, right? You could eat when you make these eggs, what are the 4 or the sperm? What are the 4? I said plant, so I guess I shouldn't be speaking about sperm, but when we're talking about these gametes, what are the 4 possibilities if they have big Y, big R, little r? What are the 4 possibilities that we can that these I'm going to go ahead and talk about in humans, these 4 sperm could possibly be. Right? Think about crossing over. It's random. So you can get big R, we can have big R with little y and big Y with little r. So there's all kinds of possibilities, but really there's only 4. And the reason there's only 4 is we can do something called FOIL. We can factor this out, right? So just gotta follow an order. Big Y, big R, little r. Let's go ahead and do it together. So if we do this, we see that big Y could be with big R. Well, that's one possibility. So that's what would go here. So that's on this chromosome, big Y and big R. Then again, big Y could go with little r. So big Y could go with little r. But then little y could go with big R. And then little y could go with little r. So there's your 4 possible gametes that this male could make. The female, again, if it were in humans, you'd have 4 possibilities. You don't know which one that egg is going to be. Using the same method, because it's also heterozygous, would be big Y, big R, and big Y, little r, and little y, big R, and little y, little r. I want to mention, I did explain this to you in that hour-long video when we doing dihybrid crosses, that this method is how we would come up with the 4 gametes. But there's all kinds of possibilities, but really it only breaks down to 4 possibilities here along this edge. And so this is half. And remember, if I ask you this question, what do the 2N, what is the zygote going to have? The zygote's going to have 2 copies of Y and 2 copies of R. 2 copies of Y and 2 copies of R. Well, how's that going to happen? Well, it's going to happen because the zygote is going to take the sperm from the father and the egg from the mom and combine the DNA. So you'll have 2 Ys and 2 Rs, but the question is which 2 Ys and which 2 Rs. That's where the Punnett square comes into play. We call this a dihybrid only because there's 2 genes involved. And in the past, you only do 1 gene. That's how we train you. We show you how to do a cross with 1 set of genes where you get 1 out of 4 chances. But when you're dealing with 2 genes, could you have yellow and wrinkled, yellow and smooth? In this case, let's take a look at the possibilities here. Here we have yellow and round. 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 So everybody here is yellow and round. How does that make sense? Well, these are dominant. So here you have yellow and round, yellow and wrinkled, yellow and round, and yellow and wrinkled. How's that possible? Well, this is dominant for this. Everyone in this row is going to be dominant for yellow, but it also has a recessive for the wrinkled gene or the smooth gene. Here you have green and round, and green is recessive, so you have yellow and round, green and round, and green and round. And the last, of course, is recessive for both green color and wrinkles. So here you have heterozygotes, yellow and round, yellow and wrinkled, green and round, and green and wrinkled. So what are your percentages? What are your phenotypic ratios? You get 9 to 16 yellow and rounds. You can count them. You can go ahead and count them. You can say, how many of these are yellow and round? Phenotypically, right? So phenotypically, you have yellow and round, this homozygous, right, dominant. This is also yellow and round, but it's homozygous for the color, but not the round. Here's yellow and round. Here's yellow and round. Here's yellow and round. So you can count them. 1, 2, 3, 4, 5, 6, 7, 8, 9. There's your yellow and round. 9 out of 16. 1, 2, 3, 4. 1, 2, 3, 4, 4 and 4, 16. So there's 16 possibilities. Only 9 of them are yellow and round. That's phenotype. Phenotype is what they look like. In genotype, big Y, big R, heterozygous, for big R, no matter what else it could be. When you're talking about recombinants, this is the recombinant genes down here. Recombinant means after you mix them up, right? So you can get what are the possible outcomes after you've done crossing over. And after you've had sex, after you've had each of the zygotes, each of the babies here. So what we're dealing with is a 1 out of 16, 3 out of 16, etc. for phenotypic ratios. If we did genotypic ratios and we say how many big Y, big double Y, double big R, double big Y would we have in this, all 16 of them, I think you can agree with me that there's only one of those. So it's 1 out of 16 that we would get out of the progeny for this genotype. What about this genotype, big Y, big R, little r? So homozygous for color, but heterozygous for smoothness. There's only 2 of them. And this one that's big Y, little y, big R, there's only 2 of them. Homozygous dominant, homozygous recessive. You'll see there's how many do you see here of this type? There's only 1 of those. And then here there's 2. Big Y, big R, little r. So 2 heterozygous combinations. Here's heterozygous for color, but homozygous for recessive for wrinkled. Here's homozygous recessive for color, green, and smooth, homozygous dominant for smoothness. Notice that these 2 are opposites. And then this one here is homozygous for color, or homozygous recessive for color, heterozygous dominant, heterozygous for smoothness, and finally you get your homozygous for color. It's homozygous for recessives for color and smoothness. And you'll notice that there's only 1 of these as well. So 1, 2, 1 to 2 to 1 to 2, etc. So you could do your own genotype depending on what they ask you. What is the genotypic ratio? Well, they have to ask you to what? And once you know that, then you can just start listing them out. It's 1, big Y, big R, big R. And it's going to be 1, little y, little r. And you can keep doing that across the board. I'm not going to write all that out, but you can see that there'd be a good combination of genotypic ratios of 1 to 1 to 2 to 1, depending on what it is you want to talk about. And that's how you do it. So where did we choose these? Where did these come from? They came from this FOIL method. This FOIL. It's an acronym. It's not FOIT. It's an acronym. So the FOIL method is just this idea of first, when you're talking about FOIL, you're talking about first, then outer, then inner, and then last. And in math and algebra, you use FOIL as a mnemonic to remember the method of multiplying binomials. And in essence, that's what we're dealing with here is a binomial. And you can go ahead and multiply these out, and you get these 4 possible outcomes. We're talking about 2 different variables and 2 possibilities, and the question is, what are the possible combinations? And just by doing it in order, it really doesn't matter which order you use, as long as you don't end up repeating yourself. You can come up with these letters on the sides, which is the question your colleagues have asked me about. Okay, how do we get these in order to find what goes inside the boxes? Now let's go ahead and use one of the other examples they asked me about. So this is a wiki that they found. These 2 people are studying together the way I asked you guys to. I hope you guys are studying, have a partner, have someone who's helping you out. Also, you have the videos, and of course we'll hit this together when we get back. But if you're not working now, you're going to be behind when we get back together in January. So here you see the parents, and what do the parents look like they are? And remember, we could use any letter for any gene. let's remember that. What are the parents? I hope everyone said heterozygote. And what does heterozygote mean? It means they have two different alleles. So when they have copies of their genes, no matter how they come up, and I'm going to go ahead and draw one now and say that, they might have, maybe they have a big A and a big P on one is when they start off, and on the other one they have a little a and a little p. But because of crossing over, you can have all kinds of combinations, including once you cross over, you have big A with little p, big A with little p, and you have, of course, the opposite, which is little a with big P, right? So you got all kinds of combinations that you can give, right? You can give the big A, big P. You can give the little a, little p. You can give the big A with big P, or the little a with big P. Any one of those could happen. And both parents are the same thing after meiosis, right? And I'm going to say it again. And after meiosis, what we get is we get these 4 cells, and this is only one chromosome, right? But in humans it'd be 23, and other organisms would be another combination. But you get these 4 chromosomes of this gene. These chromosomes have been mixed up because of meiosis, and we just don't know what it's going to give to the zygote when this gamete and the other gamete from the other parent come together. We just don't know. That's where we get these letters from. There it is. That's where we get those letters from. So when we're making our cross, we're talking about 2 different genes, where in a regular Punnett square, you're only dealing with 2 different alleles, 1 gene with 2 alleles. Here we're talking 2 genes with 2 alleles each. So now how do we draw it? We would draw, there's going to be 4 possible gametes. You see that, right? There's 1 possibility here, another one here, 3rd here, and a 4th here. There's 4 possible gametes. So we say 1, 2, 3, and 4. And on the side we have 1, 2. I'm running out of space. It's unfortunate. So I'll draw it smaller. So we have 1, 2, 3, and 4. And here we have 1, 2, 3, and 4. And so when we draw these, of course, we're going to draw a box so that we know each box is going to represent a zygote, a baby, right? If it develops completely, a baby, right? But it's a zygote at first when it's just a single cell. So, okay, so here we go. Here's our zygote. But what were the gametes that came together? What are the possible gametes that can come together to form each one of these zygotes? There's 16 possibilities here. We're talking about two genes, 2 alleles each. There's 16 possibilities. Well, it would be, we could have, let's just go down the line, right? Here, they listed them out for you already, but we can just go ahead and follow what we created here, and it should be about the same. It could be big A, big P. Well, that took care of that. So we can get rid of this one. This one's gone. And it could be big A, little p, big A, little p. So this one's gone now. So that was the possibility. So that possibility could be there. Or maybe it's the little a, big P. Maybe that's the one that made it to the egg. Big A, little a, big P. That after the crossing over of it, we mixed them all up. And then, or it could be maybe it's a little a, little p. And that takes care of this possibility here. So there's all 4 of your possibilities. And of course you would do that. The same thing would happen with mom as it happened with dad. Big A, big P, and big A, little p, and then little a, big P, and then little a, little p. Now, here's the question. Why are there 4 possibilities inside each of these gametes? Remember, every one of your cells in eukaryotic organisms, in most eukaryotic organisms, every one of your cells has 2 copies of each chromosome. 2 copies of each gene. So when we're done, each gene has to have 2 copies, one from mom and one from dad, right? Each gene has to have 2 copies, one from mom and one from dad. One from mom and one from dad has to have. So if I erase these and make them pink, so maybe that'll help you visualize this. One from mom, one from dad. Now here we're talking about 2 different alleles for 2 different genes. So that's maybe confusing you, but no matter what, you have to have 2 copies of each gene in each of your cells. Yes, I know, except for the Y chromosome, that's true. And even the X chromosome for that matter. The sex chromosomes are different, but every autosomal chromosome, there has to be 2 copies of each gene. So you get, in this zygote here, you have, what is it going to get in this probability? This is all probability, right? We don't know what's going to happen because we don't know which one of these 4 is actually going to make it to the egg, and which one of the 4 possible eggs, which are going to be the same as these, are going to match up with this, whichever one of these 4 sperm. So let's go ahead and make that assumption that we get, it could be, let's look at the probability of getting it. We get 1 here, that's a big A, and then we'll have the big A here, that came from this here, from this mother. And of course, this sperm will have a big P, and this will have a big P as well. So this was homozygous dominant. There's only going to be one of those. And there's only going to be one of the homozygous recessives. So we know we're only going to get 1 A here and a A here. And we're only going to get 1 little P here and 1 little P here, right? Because it's the only ones that are both recessive. So that's telling you something about genotype, right? All right, so then we come down in the next column. You can go any direction you want. Just come across and come down. It's just a chart. So in the next one here is a big A and a big A. And then, of course, you have a little P. You have a little P from dad, and you have a big P from mom. We usually put the big letter first. All right? All right, so then as we keep moving, you say we get a big A from mom and a little A from dad. And we say you get also a big A from mom and a big P from dad. All right. Oh, sorry. Eeks. There'd be 3 copies of A, and you'd have some trouble there. So we'd need a big P from mom and a big P from dad, right? So here you have a heterozygote A, and here you have a homozygote dominant P. Now that goes back to that terminology, heterozygote. What does that mean? That means that there's both 2 different copies of the allele. Homozygote, what does that mean? 2 of the same copies. So if you see 2 big P's, they're the same alleles, the same gene sequence, right? GTCC, CTG, whatever. They're the same exact gene sequence, same allele. And where if it's heterozygote, you have 2 different alleles. There's a difference between these 2. Some mutation in a long past ancestor gave you 2 different copies of allele. One is P for short and big P for tall. I'm not sure. Whatever the letters might mean. And those have to be given to you. There's no set variable for each trait, okay? So when we look at mom, of course, this whole row, mom is going to give all big A and all big P, right? The only one that's changing in this row, the only one that's changing is dad's, right? And if we go across in rows, that would be the same. Every time we draw these in this row, they're all going to be big A, big little P. And if we went down in columns, then only moms would change, right? Because it's just the nature of filling in a chart. All right, so dad's would be little a, little p, and I'm just going to quickly do the rest of these. Mom is going to be big A, big A, big A, of course, because that's all this row is dealing with. And she's only going to give little p across the board. And then dad is going to change up, and dad's going to give big P here in this one. Oops, I forgot the A. Dad is going to give a big A here and a big P. He's going to give a big A here and a little p. He's going to give a little a here and a big P. And he's going to give a little a here and a little p. I guess it's kind of running out of room. I'll just put it there. So that's that row. And I can do the same for the next rows. And little a, little a, little a, because I know Mom's not going to change across this row. And do that. And Dad is going to do big A. You know what? Try to stay with the convention and draw. Not that it really matters, but I like to draw the big letters first. Big A, little a. And again, this is big A. And Dad's going to have a little p. And here we got little a, big P, and little a, little p. And Mom finally is little a, little p, little a, little p all the way across the board. And Dad is going to have big A, big P, big A, little p, little a, big P. And how did I get that? How did I get these? One more time and then I'm done. Where did I get these? Each of these? I got them from the possible gametes. How did I figure out those possible gametes? I thought about it and I said if they crossed over, I can get this and this and this and this. How did I get the possible combinations? All you got to do is take, I'm going to go ahead and draw it out here. All you got to do is take Mom's big A, big P, little p, and do FOIL, right? So your first, we go A goes with P, that's your big A, big P. Then A goes the outer one, right? That's big A, little p. Then the little one can go with the big one. So the inner goes next, right? FOIL. That's little a, big P. And then finally, the last, just a, little p. That's each of these. That's where they came from. This FOIL method of taking the possible combinations that could lead to these 4 possible gametes. I hope that helped. Any other questions, please feel free to email. If this is confusing, still, maybe I can find a video that'll explain it. I don't think there's any other way to explain this. You have to kind of work it out and work out the problems and use the FOIL method, as I say to you. Use FOIL. First, outer, inner, last. You learned it in algebra. Use it here. All right. Good luck.